I'm very pleased to introduce our next speaker. Jennifer Listgarten is a professor in electrical engineering and computer science and the Center for Computational Biology. She's a member of the steering committee for the Berkeley AI Research, Bear Lab, and a Chan Zuckerberg investigator. From 2007 to 2017, she was at Microsoft Research, and before that, earned a PhD in the machine learning group at the University of Toronto. Her expertise is in machine learning, applied statistics, and computational biology. She's interested in both methods development as well as applications of these methods to enable new insight into basic biology. So, her talk today is on how machine learning can be leveraged to improve CRISPR gene editing, and how to accelerate the design and optimization of proteins with machine learning techniques. Please welcome Professor Listgarten.
It's a pleasure to be here. So I'm going to tell you that, at least from my perspective, which is a machine learning perspective, modern day molecular biology can be thought of a lot like data science. And so my expertise is in machine learning, and I like to combine these things. And what you see here is a graph of the cost of sequencing and how it's changed over the years on the top. And you can see in comparison to Moore's law just how quickly this is moving. And this is also, well, I guess it's a little bit out of date, but the trend grows. And correspondingly, we're getting a huge growth in this kind of data. So right here, what's depicted is sequencing data. But in addition to that, we're getting all kinds of new kinds of data. And then the ones that exist are being refined and improved and cleaner and created at a larger scale. And so this means there's a whole host of very interesting problems for people who like to do modeling and have an impact on the world. So something I spent many years doing is looking at how machine learning can be used to find signal in genetics. So there's a lot of very interesting problems there. I'm not going to talk about it today, but basic setup is you collect, for example, the people in this room, but ideally a much larger set of people. You collect some blood, you measure their genetics, and then you ask questions about what diseases or traits they have, and you're looking for the genetic underpinnings of these traits. And on the surface, this sounds like a reasonably simple statistical problem, but it turns out there's actually quite a few interesting challenges there. Another one that I will talk to you more about today is how you can use machine learning to do better with gene editing. So probably many of you have heard about CRISPR. This is a little video showing you how kind of how it works at the molecular level, where this can I play that again? Maybe not. Okay, back. And then forward. Right, so that thing coming in has to sort of bind to the DNA and pull it apart and do something. And so depending on what's there, it may do so better or worse. And you can model these things if you have enough data to feed into your models. And I will tell you more about that. And something that's become very interesting to me just since moving to Berkeley a year ago is how we can use machine learning to do protein design and protein optimization. So, and I will also tell you more about this one. Well, something that's really fascinating that a lot of people haven't necessarily heard of is how you can use machine learning to do drug repurposing. So what is drug repurposing? It says that I have all these drugs out there that have already gone through this very expensive FDA approval, and we know that sometimes through serendipity that one of these drugs gets used for something else and can dramatically change the world, but it's often just a serendipitous discovery of what this drug that it was intended to use for, thing A, and instead we can also use it for B. So the question is, can you sort of systematize this using the data? And it turns out that this is an ongoing field. And one example here is basically get like a molecular barcode, if you will, that is a signature for each disease. So this is microarray gene expression data. And then you have sort of a dictionary of these characteristic vectors for each disease. And then on the other hand, you can do a perturbation to a cell line with a drug and get its characteristic vector. And then you can kind of, in some sense, anti-correlate these. And the drugs that are signatures that are anti-correlated with the disease signatures, it stands to reason that these might be good candidates to reposition that drug for that disease. So this is another sort of very powerful example of what you can do with lots of molecular biology data with respect to drug repurposing. And so more generally, evolution has sort of created this very informative structure in the DNA, in the proteins, in the gene expression. Sort of all the data that we can squeeze out of the body has very rich informative structure if we have enough data and we know and understand its noise properties and the sort of confounding factors and things like this. And so we can use machine learning to extract that and tackle quite a broad range of problems. And that's sort of what I do through my career is I've moved around through different areas, and today I will focus on two of those, which I've already just given you a little preview of. So one is CRISPR gene editing, and the other is protein design. So the first is sort of work that's sort of largely complete, and the second one is very new work that is ongoing. Okay, so first of all, who here has heard of CRISPR? Very good. Okay, that means you're awake still. So, right, so, CRISPR, people have been studying it for decades, actually, and only more recently, I'm not sure actually the date on this one, it started to make its way into the big scientific journals like Nature, and then slowly into sort of more mainstream like The Economist and the New York Times and CNN. And then Wired made this one up. I think this is now 2 or 3 years old. So this cover article, every word on here is in reference to CRISPR. Who saw this Wired cover? Right, so what is no hunger, no pollution, no disease, and the end of life as we know it, the Genesis engine. Okay, so that's like a little bit over the top. And so after that went out, this backlash appeared on Twitter, and it's not very active now, but it was super active and creative. And so, right, so I would say sort of just like machine learning is super impactful, but it's also very hyped. CRISPR is the same. It's already changing the world, just like machine learning, but there's a crazy amount of hype around it, and I think this may have been the peak. I don't know, maybe it's ongoing. But the reality is that there's already very promising translational results for medicine here. And it goes beyond medicine as well. One can use this for agriculture, for all kinds of things. It's also used as a routine sort of method in molecular biology labs just to probe systems and things like this. So it really is touching on so many different areas of science and society. This is now an old slide. You can see there's nothing since 2014, but this list is super, super long about proof of principle things that have been done with this, largely in animal systems, although you may have heard some of the kind of crazy stuff in the news about the CRISPR baby and things like this. I'm not going to talk about that, but here are just some examples of the kinds of things you can imagine will be done in the sort of short, maybe, who knows, I won't speculate on time, but you could correct genetic defects that occur at birth, including things, for example, HIV requires a particular receptor, or it usually uses this one receptor to get in. There are actually a few people in the world who don't have that receptor by birth, and they're kind of essentially immune to it. So these are the kinds of things you can imagine engineering into a person if you could get this CRISPR system to work very well. And then there's this sort of crazy thing called gene drives, which is a way to induce super inheritance, if you will, although there's some speculation on whether this will actually work. And of course, it's a little bit terrifying. So of course, we need some ethicists as well. So, right, so what is gene editing? Typically, when you hear of editing, you think of like a sort of copy, cut and paste, right? You're going to change an A to a T in the genome or something like that. But it turns out a really big use case for this is actually just preventing a gene from working at all. So we call this like gene knockout. And you can imagine you would do this, for example, for drug screens. Imagine you want this thing to stop working. So what can I hit it with to make it stop working? Or another use case would be you're trying to understand like a whole sort of systems biology perspective of a cell or an organ or something like this. So you want to probe it with perturbations. And one very powerful perturbation is to stop something from working and then ask, how does that affect things? So I'm going to talk about my work with the Broad Institute, in which we were focused on this gene knockout problem. So there are kind of two challenges. I mean, there's many challenges. It's a very rapidly moving field, and CRISPR gets used for much beyond what I'm going to even tell you about. But at its core, there's kind of one key thing going on, which is the ability to precisely go in and target the genome to do something. And so in this case of gene knockout, we want to go in and we want to actually cut the DNA. And then what happens is there's going to be some repair mechanism that tries to fix it and is actually faulty in a stochastic manner. And because it's faulty, with some probability, the gene will not be repaired and it will not work. However, this is it depends on all kinds of properties of the DNA and the physical and chemical system. And so in principle, imagine this is like a schematic of a gene and we want to knock it out. This is the rest of the genome. And so you can apply CRISPR at many, many places in this gene. You can say, I'm going to target it here, or here, or here, but you have to decide which one. And it turns out that some of these work better and some of these work less well. And for some genes, many of them will work well. For some genes, almost none of them will work well. And so this is already sort of a workhorse of modern day molecular biology in pharma, in academia. People want to do this. And typically, there's hundreds of places you could target it. So where should you target it? Well, if you can collect enough data about what worked and what didn't work over a suitable training data set, this is a perfect place to sort of use machine learning. So that's what we have done. But there's a flip side, right? Imagine that I have a very good model, or I've made some decision about where I will cut it. Then let's say I chose to cut it here. Then now the question is, am I going to accidentally cut the genome somewhere else, right? This is obviously a terrible problem. How terrible it is depends on the use case. If you're doing this for like an in vivo therapeutic, this could be deadly. If you're screening drugs on a cell line in a lab, then you add noise to your measurement. So, but in any case, no matter what, we really want to be aware of this and eliminate this. So there are two ways we can do that. As I said, it's a very rapidly moving field in molecular biology, so you can improve the laboratory methods. But I'm here on the stage today because I'm going to tell you about how you can use machine learning. I'm just going to give you a feel for it. There's actually a whole host of quite interesting problems if you dig down, but I just want to give you a sense. And also, I think one of the most fascinating things about working at this intersection of biology and machine learning is the biology stories are really quite interesting. And so I don't know, does anyone here know who discovered or what company discovered what CRISPR actually does? So for decades, sort of molecular biologists were studying the system, and they didn't actually know what it was. They just saw these weird repeats in the genome. And in 2007, Danone, the yogurt company, and the researchers there figured out what it was doing, these weird repeats in the genome. And they figured it out because it really affected their bottom line, because it has to do with a naturally occurring immune defense mechanism. And so some sort of vats of yogurt would get infected and others wouldn't. And so that's when it was understood what these things that were observed in the genome were doing in their native habitat. And you can see just a few years later, people identified these key components that would enable people to do this general purpose gene editing. And the key components are basically some scissors and a way to guide them to the right part of the genome. And so that's sort of very rapid progress. But what does it look like in its native habitat before it's been co-opted? So here's a schematic of a bacteria that's been invaded by a virus. And this is the viral DNA. I'm going to use the terms DNA and RNA here. Let's just call everything DNA. And so imagine it gets infected. Then what happens is these sort of scissors, these Cas9 is a protein, can cut it out, and they can store it in its own genome. And this is sort of like yours and my adaptive immune response, right? We have these sort of memory T cells where if we've been infected once, we can kind of store a pattern, and then we're like primed to recognize it much more rapidly and combat the invasion at a later date. So this is its version of an adaptive immune response. What it does is it takes a snapshot of the DNA, and it stores it in its own DNA. And so I call these sort of viral memories. And so how does it delineate what's its own sort of functional DNA versus these weird snapshots? It puts these weird repeats here. These are actually where the name CRISPR comes from. So I think I have it on the last slide. It's clustered, regularly interspaced, short palindromic repeats. So that actually refers to these things here in its native habitat. So how many people knew that before today's talk? Wow, okay, I saw a bunch of hands, great. And as I said, now you have these key ingredients, which is you need to design you don't wait for it to happen by accident with this memory, you design the memory, basically. We call this an RNA guide because it's going to guide the scissors to the part of the genome that we want, and it's going to make a cut. And then we can then sort of invoke various mechanisms to either try to have it knocked down or to repair it with to change an A to a T. But the key ingredient here is to precisely cut the genome. So let me just tell you a little bit about this. And actually, I won't have time to tell you about this one. This one actually has more sort of interesting machine learning and statistical challenges. But in this case, as a machine learning problem, it's actually relatively simple to set up. And so what you do is you need to collect, as we do in supervised machine learning, pairs of inputs. In this case, that is the guide sequence here, which is going to target a specific part of the genome, or ideally a very specific part of the genome, and how effective it was at knocking out the gene that we wanted to knock out. And so we're going to collect a bunch of these pairs, and as we do in machine learning, the goal is to infer some function that is going to make good predictions for data we have not yet seen. Right, so it's the sort of standard setup in supervised machine learning. And so one of the great difficulties here is actually how do you collect this data? What are the properties of this data? And it turns out that's actually a very difficult problem that brings some very interesting machine learning challenges that I won't have time to talk about today. But roughly speaking, again, this is sort of the standard setup, is how do we actually encode these DNA into vectors that can be absorbed by machine learning approaches? What kinds of models should we use? And as I mentioned, this idea of getting these supervised labels is extremely challenging, and we were lucky to work with John Dench at the Broad Institute, who's an expert in this area. So once you've built such a model, then you can also start to ask questions about what were the important features. And so this is not a sort of very precise line of reasoning, but it gives you a sense of what's contributing to the model. And so one thing that was quite interesting was this, supposed to be the same video, is this idea that thermodynamics of the system play a role in whether you can knock down the gene. And so why thermodynamics? As I said, this Cas9, these are the scissors, they have to come in, and if the video were running, then it would pull apart the DNA. And so pulling apart the DNA requires energy that's sort of proportional to the thermodynamic setting there, which in turn is encoded by the nucleotides, by the structure, and all these things. And so unlike in a lot of the areas you hear about machine learning blowing away benchmarks with deep neural networks, which is namely vision and speech, there they have lots and lots of training data, and sort of deep neural networks can create the features that it needs because they have sort of almost unlimited data. We have very limited data here, so we're still in a scientific domain where expert knowledge can help us. And so working very closely with John, he gave us he looked up crystallography experiments about how this thing comes in, how it binds, and uses these kinds of ideas to help us find features that will be useful. So, at the end of this, we ended up with a state-of-the-art model that was absorbed by a couple of startups, and we also have, we pre-computed for the whole genome all the on-target scores. That's this thing I just told you about, is how well will it work to knock out this gene. And the thing I didn't have time to tell you about, which has a lot of interesting machine learning, this off-target business, is am I going to create accidents somewhere else? And the accident somewhere else is actually computationally a very challenging problem because you don't have to consider just one area on the genome. You now have to say, is there an accident anywhere along those 3.2 billion nucleotides? And so just even the logistics of scanning through this, let alone the statistical learning, is very challenging. And so at the time I did this work, I was actually working for Microsoft Research, and we pre-populated this database so that people could use it without having to wait, as they had to for the other tools. And so we actually, to populate this database, we used 17,000 cores for a month straight. And we did that 3 times during the revision of the paper. So, great. Okay, so that gives you just a little bit of a sense of how machine learning and gene editing can play together. And now I'll tell you something much more new that's less well developed than that, related to protein engineering. But first, I'm going to show you this slide. So has anyone seen this picture? Does anyone have any idea what's going on? Okay, so there's a dire warning put out by the gallery in Paris that it might take more than 2 days, which was as long as the exhibit was scheduled for. So this is actually an art exhibit. So I'm not going to tell you about art, but what I'm going to tell you about is that there are problems much like this, where the consequences are more dire than that the guy in the gallery does not find the needle in the haystack. And so really, I'm going to tell you about is how can we think about using modern day machine learning and molecular biology to search through protein and molecule space in order to do protein engineering and protein optimization. And so just to give you a few examples, you can imagine that something that's very important is how a protein folds will dictate if it's stable or not. And these days we use proteins for drugs for many things, and typically you want them to be stable. And it's only been in the past few years that people have been able to create synthetic proteins that were stable. This is an extraordinarily challenging problem. Sometimes you want to just make something more stable, and that's going to depend on how it folds, which in turn is dictated by its sequence. And so you can imagine that you're searching through some massive sequence space to try and achieve some particular stability that you're interested in, maybe a maximal stability, maybe above some threshold or something like that. And another example, just to link this back to my previous talk, is this thing has to come in and bind. And so while I've been telling you about how you choose which part of the genome to get it to bind to, separately people are actually engineering these scissors to get them to have different properties that they care about. So for example, maybe to bind better or not to have as many off-target properties. And so interestingly, as we've been pursuing this, I've also been approached now by several chemists, well, actually some chemists in pharma and also at Caltech, organic synthetic chemists. So it turns out the abstraction we use in a sort of statistical machine learning sense is also mostly readily deployable to help organic synthetic chemists look through chemical space. And so, right, so but back to just the protein space, for example. So proteins are composed of the alphabet of amino acids. And so if you have just something that's only 5 amino acids long, which is not a protein that really exists in nature or does anything real, you already have this many sequences. And so, 100 amino acids is not even a big protein still. And yet you can see how many possible sequences you can get at that length, right? This is just like anything beyond what a computer can do in terms of enumeration. One has to be very, very clever. And so, and actually this is a nice reference point, which is there are this many grains of sand in the world. And actually a few weeks ago when I gave this talk, the speaker before me had this exact same number. And he apparently said this is the number of seconds that have elapsed since the Big Bang. And so I feel like there's some sort of weird conspiracy going on. But I guess interesting facts, right? So how can we use machine learning to search through this molecule or this protein space more effectively? So let me give you a little bit of a flavor of that. Here's an example of a protein that fluoresces. This is a very, very common thing that's used all over the world. It's actually a big industry because it can tag things that you can see. And it's also just a nice canonical system you can use in developing methods for optimization because you can easily measure the fluorescence of a protein. One of the difficulties in a lot of these fields is in biology, you cannot really measure what you want. So you have these proxy measurements. And so if you're interested in developing computational methods, it's sometimes nice to start with a system where you can actually measure the thing you care about to see if the computational methods are working. So what is the setup here precisely that I'm talking about? So I'm going to tell you that we are going to assume we have some sort of a predictive model that goes from an input space in which we want to do design to a property we care about. And in general, we can do this for multiple properties, but just for the simplicity of this presentation, I'll tell you about one. So we're going to assume we have this model. So these kind of, commodity machine learning in some sense to build predictive models if you have enough data, if the data is high enough quality, and if the problem has enough signal in it. And that's not always the case, but I'm not at the moment interested in constructing these. So the starting point will be I have a predictive model that goes from my input design space to the property I care about. Maybe I want to optimize this. Maybe I want to specify it. And what I want is I want a method that's essentially going to, in some sense, reverse it. I'm going to specify the output, and I want it to tell me what the input should be. So this is what I call kind of sequence design, input design, or protein design in this particular case. So, and I may also have some constraints here. So I may not arbitrarily be able to change the amino acids. Maybe I can only change the DNA that underlies the amino acids in which there's a redundant encoding. So in other words, for a given amino acid, there's still a combinatorial, given protein sequence, there's a combinatorial explosion of ways this can be coded in the DNA owing to redundancy in that encoding. And so I may want to constrain it to be a particular set of amino acids, or I may say, okay, I'm willing to change the protein, but I want it to adhere to certain 3D structure that I'm aware of because I know I don't want to change that, but I still want to optimize it for other things. Okay, and most importantly, I said the starting point is that I want this mapping here as a starting point. And more importantly, I want this thing to be just what we like black box, if you will, which I don't know if the EE folks use this term as well, but what that means is I don't need to know its insides. I can't necessarily compute derivatives on it. And the reason that's important is I want to be able to sort of grab domain knowledge from wherever it is in whatever form it exists, and that may not be a model that I can peek into. And so we want sort of derivative-free methods to do this optimization. Okay, so I've told you what I want to do. Now the question is, how do people do this stuff right now? So interestingly, when we put our preprint up was the same day that Frances Arnold got the Nobel Prize in Chemistry. And in a sense, what we're trying to do is an in silico version of what she has won the Nobel Prize for. Or also you could say that what we're doing would act synergistically with what she has won the Nobel Prize for. So what is this? It's called directed evolution. And it's sort of self-explanatory, but I will nevertheless explain it to you. So imagine you have this protein fluorescence issue, and you start with some protein, and maybe it fluoresces already a little bit, and you say, but I want it to fluoresce much more, or I want it to bind to this target much more, whatever it is that you care about. And so what you do is you apply a diversification step in the laboratory. So that could be something like throwing X-rays at it to induce random mutations. There's other forms of inducing sort of random changes. But the key here is they're almost always random or very stochastic. You can tailor it a little bit more, but basically you think of this as a random diversification step. Now you have a pool of variance from the thing you started from, and now you need to, again, depending on the property you care about, you need to be very clever in the lab to measure something that correlates with it, some proxy to that thing, to see how well each of these variants does. And then what you do is you direct the evolution. So what does that mean? Well, you're declaring the fitness function. It's not my survival or my childbearing. It's the protein fluorescence or protein binding. And then you take the top performers and you start again and you cycle through. So slowly what happens is you're shifting a population of sequences through a cycle that more and more adhere to the properties you care about. So that's how it's done now. And as I said, what we want to do is a sort of in silico directed evolution. And the first obvious thing you would do is you would replace the screening step with this predictive model. And that's sort of what I said was an assumption going into our methods, is that someone has given us such a model. And that can be an interesting problem in its own right. But right now I'm sort of super interested in how can we make the search smarter? Because what kind of a search is directed evolution doing? It's basically doing like a random greedy search. And for people who know anything about optimization, this is sort of not likely to be a very effective search. And so it stands to reason that we could do dramatically better than this, and also, of course, work interactively with the wet lab acquiring new data. Right now, the work is focused on just saying we have a snapshot of the data. Let's do our best to leverage that to find the protein that we want. So I'm just going to give you a little bit more of a flavor of how this works. We take, again, a normal predictive model. Here it's not a black box. This is just some neural network. And again, normally for neural networks, you give it the training data, right? The inputs that are paired with the outputs, as I told you about before, and you tweak these knobs using various optimization schemes to get a fixed predictive network. And the use case is that you then give it unseen inputs that it didn't train on, and you ask it to make predictions. Now we're going to turn this on its head, and we're going to have a list of properties we care about. We will either say we want to maximize it, we want it to be above some threshold, or we want to specify it. And we want to be able to handle all of these simultaneously. And so one thing that's going to be really important here, and is a very open problem in machine learning these days, especially for the most powerful models, which are deep neural networks, is understanding the predictive uncertainty of these models. So they're known to be overconfident, they're known to extrapolate very badly, and so again, there's just so many interesting problems here about how we can deal with this. And we have some methods now that I won't tell you about, but we definitely want to think about how this uncertainty plays out. Does the model, you model the uncertainty, is that uncertainty correct? How does it relate to the accuracy and things like this? So we have a very nice sort of formal mathematical development of our approach to do this, which I won't go through today. But what I will tell you is what emerges from this nice formulation is a very natural and intuitive algorithm, and it has a direct mapping to directed evolution. So roughly speaking, we're going to end up with, these are the inputs I told you about. You can just lump these together. This is this predictive model that goes from input to output. This is basically the mean prediction, and this accounts for the noise. And then what's going to replace the sort of X-ray diversification step is what we call a generative model. You can think of the simplest generative model as like a Gaussian. A Gaussian has a mean and a variance. As I change those knobs, it's going to generate different data, right? Modern day machine learning, we have much richer model than a Gaussian. It has its own knobs. And this thing, we're going to draw samples from it, and that's going to be how we diversify our variant pool. And then, so we can initialize with a set of proteins or not. And each one is going to get a weight. And now we're going to do something cyclic. So there's going to be a for loop. And what we're going to do is we're going to induce this sort of mutations and variants by, first we'll train that generative model. So think of like a Gaussian, but fancier. We're going to train it on either the initial set of proteins or some randomly generated set. And now that's sort of the first diversification step, is we draw samples from it. And then instead of measuring in the lab how well each of those variants performs, we're going to score it with our predictive model while accounting for the uncertainty of that predictive model. And based on how well it does, it's going to get a weight. And so we're going to actually use all of the samples. Unlike in directed evolution, you just use the top performers and they get equal weight. So what emerges here is a more nuanced scheme. And then we're going to retrain that generative model where the samples are weighted corresponding to how well they adhere to the properties we care about. And so as you go through this, you get the same effect as you get in directed evolution, and it should, depending on how good that predictive model is and our understanding of its uncertainty, we should be able to do much, much better than in directed evolution. And it turns out, unbeknownst to me at the time I started this, that the techniques, the sort of machine learning techniques under here have very close ties, in fact, to reinforcement learning in robotics and also to image generation in vision. So I'll stop here. I think that's enough to just give you a feel, but I'll say that this is early stages, and we're now pursuing this in various collaborations with Caltech, with a pre-competitive pharma consortium, and some folks on campus here who do directed evolution, and we're hoping to have like an end-to-end story, hopefully in the next year. And I'll stop there and just acknowledge my very first PhD student when I arrived here a year ago. David has really been driving this, and Hanbom at UW, who does protein stability. And the gene editing was joint work with Nicolo Fuzzi, who's still at Microsoft Research, and John Dench at the Broad, and then a large team of people who really made that work. Thank you. I don't know, is there time for questions? All right, I see one there.
With your collaboration with the pharma, do they already have a predictive model for you to use?
So that collaboration just started a week ago. They have all kinds of, they're actually trying to build a whole pipeline. Basically, each pharma is not willing to invest in this because it's sort of too pie in the sky for them. So they managed to get some money and a team of about 10 people together to do this. And they do have predictive models out there. The question is how good are they? How well do they extrapolate? I mean, just because they're there doesn't mean we're not going to ask questions about them. So in fact, even though I kind of glossed over that, we care deeply about that and we'll do our due diligence to understand the models that are there. Many models exist for many properties. How usable they are, how well they work away from the domain they were trained on, and all these things are things we need to look into.
You mentioned the random greedy search algorithm being used or by nature or whatnot. How is that determined?
So I mean, there's two steps there. One is that the experimenter does not have a lot of control over, has some, is how you induce that variation. So if you just hit the protein with X-rays, it's just going to sort of randomly mutate. There are kind of more clever ways now where you can take a protein, you can kind of cut it up into chunks and choose to sort of plug in, you would start with a bunch of proteins and put each one into sort of Lego blocks, and then you can mix and match the Lego blocks, and you could try to do that in a way that it maintains certain structure or things like this, but it's still a very random process, even if you do something a bit more clever. And then the second component is that how you choose to retain the samples that go into the next iteration. So the way people retain them is they always retain the top X performers. But as you may know, if you do sampling and things like this, it's often advantageous to find a global optimum by not always taking the top performers, right? And so you'd really like something that's like you can write a derivation down in sort of statistical formulation that that leverages ideas from combinatorial optimization and all kinds of other things.
Thank you very much.